On personalized medicine, and personalized medicine, maybe to say beforehand, it's an approach. It's a new approach to health and care. And some people say, "Oh, we know that we always had it," but I think we hope that we can convince you that there are really some fundamentally new things. Characteristics of personalized medicine. So it's not just a buzzword, but for example, in Europe, we prefer the wording personalized medicine, somewhere else it's precision medicine, then we have stratified, etc. So don't let us get into these semantics, but as a new approach and to see where is something fundamentally new. And we will see that nanotechnology and nanomedicine plays a central role as an enabler for enabling technology and also analytics for personalized medicine to drive the way. And when I look, CLINAM is clinical nanomedicine and targeted medicine also. So it is already in the title, and now the question is to what extent? So we will have 5 speakers with very different backgrounds, covering many aspects of personalized medicine and nanomedicine. For the CVs, please have a look in the booklets. There are more extensive CVs. And the first speaker is Professor Dr. Andrew Owen. He's Professor of Molecular and Clinical Pharmacology at the University of Liverpool. He's chair of the British Society for Nanomedicine, also affiliated to the Wolfson Center for Personalized Medicine. And for his focus of research is the interpatient variability in pharmacogenomics, pharmacokinetics, and the translation of nanomedicine candidates to the clinical application. So he will bridge, I would say, nanomedicine, pharmacology, and personalized medicine. And then we have Professor Dr. Hans Lehrer. He's a former director at the Max Planck Institute for Molecular Genetics in Berlin. He's a founder of Alacre Theranostics and the Dahlem Center for Genome Research and Medical Systems Biology. And he is a pioneer of the virtual patient model and coordinator of the new and starting soon EC flagship Future Health. And he will bridge genomics and these virtual models and personalized medicine. Then Dr. Ansgar Hebborn, he's the head of Global Health Technology Assessment and Payment Policy at Roche here in Basel, and he's member of many, many national and international pharmaceutical policy fora. He's, for example, the vice chair of FBPhS working group on health technology assessment, active member of UNETA, the European Network on Health Technology Assessment, of the Health Technology Assessment International, of ISPOR, etc. And he's bridging health technology assessment, pricing, reimbursement of innovations, personalized medicine, needs-based personalized medicine. And then Dr. Inge Herrmann, she is a group leader of Particle 3D at the Department of Materials Meet Life at EPMA in St. Gallen. And EPMA, you know best, is the Swiss Federal Laboratories for Material Science and Technologies, and having a background in biomedical engineering and also clinical trial management. She has been the pioneer of nanomagnet-based blood purification technologies. And then I myself, I'm a professor of public health genomics at Maastricht University with background in clinics and pediatrics, public health at governmental bodies and academia, and have been with the field of public health genomics in Europe and going towards personalized medicine, so bridging more basic science and policy making. So, and we will have 15 minutes for each talk. We have to be very strict with the timelines, 10 minutes plus 5. So there will be time for short statements or questions. So now I would like to invite Andrew for his talk. I'm very much looking forward to it, Andrew. Thanks a lot.
Thanks very much, Angela. So, as Angela just said, I'm a pharmacologist. So I'm going to give you some thoughts about where I see pharmacogenetics for nanomedicine. I'm going to start with small molecule pharmacogenetics and then build from there to speculate about what we may be able to achieve for pharmacogenetics of nanomedicines going forward. So what is pharmacogenetics? Well, pharmacogenetics is the use of genetic information within the population to stratify patients according to either pharmacokinetic differences in drug exposure or pharmacodynamic differences in terms of drug response. For the purposes of my presentation this afternoon, I'm going to focus on genetic variability in drug metabolism enzymes and drug transporter proteins, really as trying to get predictors of variability in pharmacokinetics, with the ultimate aim, of course, of being able to identify patients where appropriate pharmacokinetic exposure can be achieved. So really, from my perspective, the pharmacogenetics is about identifying the right drug for the right patient at the right dose. So what do we know about small molecule drug metabolism and pharmacokinetics, or DMPK? Well, we know that the critical proteins involved in DMPK are the metabolic enzymes and the transporter systems. The cartoon on the left here just shows the bloodstream in pink. We have the intestine here, which has a whole cluster of drug transporter proteins and drug metabolism enzymes. So any orally delivered drug needs to traverse this barrier of transporters and metabolism enzymes in order to get into the systemic circulation. Once in the systemic circulation, or for example, if a drug is parenterally administered, then it's going to be influenced by transporters and phase 1 metabolism, phase 2 metabolism enzymes, predominantly in the liver, but also in the kidney. And we also know that anatomical compartments are very well protected by transporter systems. For example, the blood-brain barrier has a high number of drug transporters. So how do these things work? Well, if you get inside the hepatocyte here, we know that drugs enter the hepatocyte via active uptake systems on the basolateral membrane. Once in the hepatocyte, they're metabolized. They also get effluxed out into bile via efflux systems. And these things are regulated by a complex network of transcription factors, most commonly studied things like the pregnane X receptor of the nuclear receptor family. And these things bind to the enzyme or transporter gene promoters and alter the expression at the mRNA and protein level for these proteins. So genetic variability for small molecules comes from transport systems, comes from metabolism enzymes, and it comes from some transcription factors. So over the past 15 years, my team has been looking at genetic predictors predominantly in HIV therapy, and there's just a few examples here. Just a few examples here for a drug metabolism enzyme, for a drug transporter system, and for a nuclear receptor here, showing the influence of polymorphisms within these genes on the pharmacokinetics of these drugs. So those are the mechanisms for small molecules. If we consider try to put size in perspective for this, we know that a standard bilayer is around 4 nanometers in depth. A typical transport system is around 10 nanometers by 8 nanometers in size. Drug metabolism enzymes tend to be a little bit smaller than this. So what does that mean in the context of a nanoparticle? Well, if we just bring in a 100 nanometer nanoparticle here into this system, I think it's probably quite obvious that a drug transporter system or a drug metabolism enzyme is unlikely to have a dramatic effect on a nanoparticle. I always say that nanoparticles are thought of as being small, but in a pharmacological sense, they're actually very, very big materials. Of course, constituents of the nanoparticle, either excipients or stabilizers or the drugs loaded within the nanoparticle, may leach out of the carrier system and be acted upon by transporters or metabolism enzymes. So one of the big advances that has happened recently in nanomedicine in the HIV area is the development of these long-acting nanomedicines. They're a single intramuscular depot of nanoparticles which give therapeutic exposure for up to 3 months in some cases. For this particular drug, rilpivirine, we've recently identified for an oral rilpivirine regimen a polymorphism in CYP3A4, which is related to exposure. Now, this is unlikely to be implementable as a clinical test, mainly because the magnitude of difference in PK exposure is very small. But if we take this difference in clearance and through physiologically based PK modeling, look at the predicted effect of this variant on a long-acting medicine, you can see that in this case you may be able to tune on an individual level the duration between administration of this nanomedicine. So for example, going from a Q4 weekly dosing to a Q5 weekly dosing and still having higher concentrations in Q5 dosing with carriers of this polymorphism. So what other mechanisms apply to nanomedicines? Well, we've already heard a lot about these during CLINAM already. One of the things I just wanted to look at in more detail was the endocytic processes, so phagocytic and non-phagocytic pathways. We know that size influences the different mechanisms which are triggered for handling of particles in this way, and we also know that these processes influences the pharmacokinetics. So I had a look at some of the genetics behind some of these mechanisms. This is an excellent review paper that came out of RIVM, which summarizes some of the opsonization pathways for nanoparticles. Now, interestingly, many of these complement components are known to have an inherited contribution since the mid-1980s, and the molecular basis for these inherited differences has emerged over the last 20 years or so. Many of these complement systems have been shown to be associated with different infectious diseases and different ideologies. And so, I think there's potentially candidate genes there for studying the influence on pharmacokinetics of nanoparticles. If we look at some of the plasma membrane receptors that mediate recognition, there's a number of examples. The toll-like receptors, for example, there's polymorphisms in TLR4, which influence infection by diseases which rely upon phagocytosis and endocytosis, such as tuberculosis or leprosy. Similarly, with the mannose receptor, there's data showing an influence with leprosy of polymorphisms in the mannose receptor. Other examples with DC sign, and again with tuberculosis susceptibility, and indirect effects, for example, with the complement receptor, where this polymorphism in CD11B has actually been explicitly shown to influence phagocytic activity within macrophages. So a lot of good ideas there for looking at the effects on PK of nanomaterials. Finally, intracellular signaling pathways. There's a couple of examples where these things have been studied. The Rab proteins tend to be very well conserved, so that you don't get genetic differences in these proteins without a profound phenotype which manifests as disease. But nonetheless, there are these genetic associations for Rab, for the Rab escort proteins, and also in terms of macrophage or monocyte effects on phagocytic activity. So just to summarize, I think it's fair to say there's very few, if any, studies which have really attempted to look at pharmacogenetics of nanomedicines. The mechanisms that underpin variability and exposure are different for nanomaterials, so we need to consider other things. I think the protein corona formation, the surface receptor recognition, and signaling pathways are good plausible candidate genes for study, but there may be other particle-specific mechanisms as well, for example, in structural proteins or in immunological genes which may influence safety. The last point I just wanted to make is even in the absence of a clinically implementable test in pharmacogenetics, a significant association in a patient cohort does give you important information about mechanisms which determine the pharmacokinetic variability in a particular medicine. So I'll leave it there. Just thanks to the team in Liverpool, everybody who funded us, and all our friends and colleagues in the UNCL.
Thank you. very much, Andrew. A short statement or question? We have time for that, please. Next step, what do you think? What is the next challenge, a big challenge?
Well, I actually think that, broadly speaking, as you know, there's two ways of looking for genetic associations. I think you've got challenges associated with both. So you've got the candidate gene type approach versus the genome-wide association approach. I think the big challenge is around understanding the mechanisms which underpin the pharmacokinetics in the first place. I think that we've got a fairly good understanding of some of the mechanisms, and that might yield good candidate genes. But I think, as in most areas in the genomics area, I think the complication is how we use the more hypothesis-generating approaches to really identify mechanisms that contribute, particularly for something like pharmacokinetics, where variability is multigenic rather than a monogenic phenotype.
Thank you very much again. Okay.